Okay, chapter 5 is all about the membrane. The class, all about the cell. Membrane, major part of the cell, one of the four main things found in a cell. And really, it's one of the defining characters in that. Shown here is Grand Central Station, which you think about it, is sort of like the gateway to the subway station or train station. Outside the city, inside the train station, if there was no walls or no gates, there would be no differentiation between in and out. That's kind of what the membrane does, and it allows passage of certain molecules, slow certain molecules. And so to talk about medicine, pharmacy, nursing, all this stuff, you really have to understand, to understand a cell, you've got to understand the membrane. Okay. So the model, and this is called the fluid mosaic model. Model is just really a way to take something very complex and make it simple such that you can wrap your head around what's going on. Okay, this is called the fluid mosaic model. The fluid part implies this fact that the lipid bilayer, here shown in yellow and red, is just that. It's not a solid. It's a fluid structure that can move, it can change, and the mosaic part of this is talking about these different molecules that are embedded into the lipid bilayer. Okay, you'll see some of these proteins that are integral. They go from one side of the membrane to the other. Other are called peripheral, meaning they're just on one side or the other. And some of these have additional modifications made. Here, glycoprotein, GLY, means carbohydrate. Glycolipid is just a lipid with a carbohydrate attached that's sticking into the membrane. And then we have, I guess that's the main one. So these molecules that are embedded we're going to learn about what they do and why they're in the membrane. Review. All right, we have the amphipathic, the polar head, hydrophilic polar head, because of all these oxygens. See, they have the partial negative charge. And then we have this long hydrocarbon tail, very nonpolar, very hydrophobic. So when put into solution, the natural behavior of these phospholipids is the heads will stick together, the tails will point the other direction. The tails do not like water, so if you just put a whole bunch of these individual molecules in water, they're going to sort of form up into a big sheet. And if the tails are sticking down in the water, they don't like that, then another sheet will fold up, and they're going to keep their tails inside of the hydrophobic heads. So it's to me, again, it's still an analogy, but it's like a bunch of buffalo or musk ox, and if they have their babies that they want to protect, and there's a lion or something, what do? They all form a ring with their heads pointed out towards the enemy and their back facing towards their baby. So it's sort of the same in my mind, at least. Here are those integral proteins. Right? We're starting to combine actually chapter 5 with chapter 3, when we talked about macromolecules and secondary and tertiary structures. So here the yellow is the bilayer, and the pink and the blue are these proteins. And again, you see these motifs. We talked about these. This would be like a pleated sheet. These might be alpha helices. The chemical properties of those make them, they would be more would make them, let's see, if they stick in with the tails, that means they must be hydrophobic or non-polar. And they just fit right into the membrane here. So these are the same as these here. Okay. The applied part of this. Okay, who cares about glycoproteins and proteins and all this? Well, here is the lipid bilayer, cytoplasm on the inside, cell on this is the extracellular matrix. This is exactly how different viruses gain entry into your cell. Okay, this is HIV specifically, which HIV is not, I mean, when I was a kid we talked a lot about HIV. Now it is all about our friend, let's see, how do we get out of here? COVID-2, SARS-CoV-2. So just out of curiosity, I pulled this up and check it out. Here is, this is a very important peer-reviewed journal called Frontiers of Cell, I don't know what it is, Cellular Infectious Microbiology, and out in October 2021. And this was a big question, was how does the SARS-CoV-2 virus gain entry into the cell? Because until you understand that mechanism, until you understand this, you can't produce any kind of therapeutic drugs. It makes it harder to develop any type of medicine for the substance. So it turns out the receptors are called ACE receptors. So here's our look at this. So I'm telling you guys, this stuff that you're learning in this class already just in the first 3 weeks will help you to understand some of this very important stuff, right? So if we look, this is the spike protein on the outside of the SARS, the COVID virus, and it integrates with this receptor. We're going to talk about receptor-mediated endocytosis in this class. This is exactly what you're seeing. It is received by the receptor. The plasma membrane forms a bubble around it. Suddenly the virus is on the inside where it can wreak havoc on your body. So, super relevant stuff. And we'll see these receptors, these little signals, glycoproteins. These are oftentimes like little flags that tell your body what type of cell this is, and they tell you, again, what your blood type is. They're on the external surface of the cell. All right, very key diagram. Get this out here. Yeah, there we go. Okay, here's the membrane. Some things on this end of the gradient. It's a permeability scale in terms of how fast, how many centimeters per second? Oh yeah. How many centimeters per second it will move through a phospholipid bilayer, these different substances. Down here it's 10 to the negative 12, so they're going to move very few centimeters in a second. And as indicated here by this bounce. Right, so these substances, Cl negative, K plus, Na plus, right, they do not move readily through a plasma membrane. Why is that? Alas, you know, what is Cl negative, K plus? What do the pluses mean? It means it is a charged ionic form of the element. Charged particles do not move through membranes. Glucose, not charged, but they're relatively large molecules. They'll move through, but not entirely easily. Things like water, glycerol. Glycerol is a little bit less likely to move through, right? But they move through. They're uncharged, but they're polar. Okay. And then when we look at these very small and nonpolar molecules such as free oxygen, carbon dioxide, nitrogen, these require no energy to go through the plasma membrane. So it's an important concept, is how to predict if I tell you a given molecule, or say it's a molecule again that's involved with some type of a drug, and scientists would be able to tell you without really setting up an experiment. They could do calculations that would tell how quickly that substance will easily move into the cell based on its size, its charge, and its polarity. So spend some time with this one for sure. So ions do move in and out of plasma membranes. Okay, molecules, big or small, do move in and out of plasma membranes. How do they do this? Okay, and this is kind of an outline of this part of the lecture. These are different methods that describe ways that these molecules and ions move across membranes. And we can think of the first two here, we call passive transport. They are free to the cell. The cell does not need to use any energy to move things by diffusion osmosis. Facilitate transport may or may not require energy. These two require energy. Okay. Let's go through these each. Another concept, it's an easy concept, but an important one. It talks about gradients. And this is about diffusion and about concentration of molecules in and around a cell. So here's the cell. We're looking at just the edge of the cell, inside, outside of the cell, extracellular cytoplasm. Through time, if we put these small, I guess they must be small and polar molecules outside of the cell, they will, just by nature, just due to physics, move through certain number of them will move through the membrane into the cell. If you keep going in time, this is going to be the stable configuration here. Why? Because we end up with an equal number concentration of the substance outside and inside of the cell. Why is this not stable? Because there's higher concentration outside than there is inside. Okay, it's like the fart in the elevator. If you're sitting over here and you fart, they're eventually going to smell it. Why? Because you see these particles will eventually diffuse throughout the inside of the elevator. Unavoidable. Okay, that is passive transport. No need for the cell to spend any energy. Other molecules, okay, these might be things, again, in that second stage, something like a water molecule, maybe a glucose molecule, probably a water molecule would be a good example. These things will move through a membrane, but not particularly easily, and they are going to be moving down a concentration gradient, i.e. from high concentration to low concentration. This is passive, however, but rather than moving directly through the membrane, there's a little protein that's in the form of a channel, a tube, and those molecules move down through the channel. Okay, so it's passive, no energy expenditure, and it's moving down the concentration gradient. Okay. Some of these things, we see the main difference here is the protein itself. Right? This protein actually has to change shape in order to transport that molecule. Okay, so it could be that this, as the molecule is received inside the protein, that causes what we call conformational change, and it moves the molecule down. Key thing though, we're still talking about from high concentration to low concentration. Okay, so this is like water flowing downhill. If you are a company that makes hydroelectricity, water is going to flow over a dam just by the same basic principle of gravity and concentration gradient. So we're going to stop it here, and we'll pick up with active transport.